Today we're going to look at how blood glucose concentration is regulated. But before we start looking at that, again, a few questions just to get you thinking. So have a go at these questions, jot your ideas down on a piece of paper, try to use keywords and as much detail as possible so that you can start thinking about the way that you might structure an answer if this were an exam question. So the questions: what does the body need glucose for and what does it turn glucose into? Why is it essential that blood glucose concentration remains constant at approximately 90 milligrams per centimetre cubed? Without maintenance, what would happen if the concentration got too high or too low? And what causes an increase or a decrease in blood glucose? Pause the video now, jot down your ideas. So hopefully you've had a chance to put your ideas down on paper. So now let's have a look at some of these answers. So what does the body need glucose for and what does it turn glucose into? Well, of course, glucose is the main source of energy in the body, and as it is required in the production of ATP in respiration. Why is it essential that blood glucose concentration remains constant? Without maintenance, what would happen if the concentration got too high or too low? So if it got too high, you would have hyperglycemia, and that would cause you to become thirsty, hungry. The long-term effects would lead to fatigue, weight loss, poor wound healing, damage to the retina, kidneys and legs. If your blood glucose level became too low, then you would have hypoglycemia, where you would end up with blurry vision, a rapid heartbeat, you would be shaking, have a headache, and if your levels dropped considerably, then it could result in you fainting, having a seizure, or even going into a coma. So that's why it's really important that that blood glucose concentration is maintained. So there are different ways that your body responds to insulin and glucagon to either increase or decrease the amount of glucose in your blood. So there are three kind of new words here that you need to know. We've talked about lysis actually before. The word lysis means splitting, neo means new, and genesis is birth or origin. And so those words appear in some of the new words that we're going to look at today, but knowing what those three words mean will help you to remember these longer biological terms that we're going to use. So to increase the blood sugar levels, there are three things that can happen. You can either eat more, okay, carbohydrates in particular, that's going to raise your blood sugar levels. We can have Glycogenolysis, which we've talked about before, which is Glycogenolysis. Remember, that's splitting that glycogen, and we're splitting that glycogen into glucose units. That's going to occur in your liver and your skeletal muscles. And then we have Gluconeogenesis. So let's break that word up. Gluco, as in glucose, neo, new, genesis, birth. So we are creating new glucose from something else, and what we're doing is actually we're making glucose from a non-carbohydrate source, such as a lipid or an amino acid. If you don't have enough carbohydrate left in your body for respiration, then your body will convert some of that lipid that you might have stored on a rainy day when you had pizza and chips. It breaks that down into glucose, so that then that glucose can then enter respiration and be used to produce ATP. To decrease blood sugar levels, there are two things that can happen. One, exercise and respiration. Obviously, if you're exercising, you're going to be needing more glucose to produce more ATP, and so that's going to decrease your blood sugar level. The other one is glycogenesis, so glycogen creation. Okay, and again, that's going to happen inside the liver. This is a nice, easy way to help you structure an answer showing how insulin and glucagon interact. And actually, if I were in an exam and a question came up about this, I might just sketch it alongside the question just so that I could refer to it while I was answering the question. So there you've got your normal blood glucose concentration at 90 milligrams per centimetre cubed in the centre. And on the left hand side we've got what happens if our blood glucose level rises, and so we have the secretion of insulin, and on the right hand side our blood glucose level has decreased and we have the secretion of glucagon. So let's look at the insulin side first, the high blood glucose level. So if your glucose level increases, then the first thing that happens is that is detected by the beta cells in the pancreas, and those pancreatic beta cells in the islets of Langerhans are going to release insulin. That insulin enters the blood plasma and will travel all around your body, and pretty much every cell in your body has insulin receptors on the outside of it. Red blood cells are an exception to this, and once that insulin clicks into place with those cell surface receptors, what it does is it causes channels in the membranes to open to allow more glucose to enter the cells. And that happens particularly in the skeletal muscle cells, which is where we're probably going to need an increase in glucose. The cell respiratory rate also increases, therefore increasing their need for glucose, and so we get a higher uptake of glucose from the blood. We also have increased glycogenesis, so the production of glycogen in the liver from glucose. That's mainly in the liver, although we do have some in the muscle cells themselves. We also have an increased rate of glucose conversion to fat, and we get inhibition of glucagon release. So we're stopping glucagon from being released. Now, all of this will therefore reduce the concentration of blood glucose and it will return back to normal. That then feeds back, once we hit that normal, that feeds back to those beta cells. Those beta cells are detecting that, and therefore we stop insulin production. So therefore we have negative feedback because we are feeding back to now reduce the amount of insulin that is being secreted. On the right hand side of the diagram now, we've got what happens if the blood sugar level falls. So we have low blood glucose concentration. So that's going to be detected by the alpha cells in the islets of Langerhans, and they are going to release the hormone glucagon. Now, glucagon is slightly different to insulin. With insulin, remember, the vast majority of the cells in the body had receptors to detect that insulin and to respond to it. With glucagon, it is only liver and fat cells that will respond. Other cells do not have receptors on the outside. So there's three things that happen. One, we get increased glycogenolysis. So remember, glycogenolysis, so we're splitting that glycogen, breaking it up into glucose in the liver to be released out into the blood plasma. We decrease the amount of glucose that is absorbed by the liver, and we also increase gluconeogenesis. Remember, gluconeogenesis is the production of glucose from lipids or from amino acids. Once the blood glucose level has returned to normal, then what will happen is that again we have negative feedback, so that feeds back and is detected by those alpha cells, and so they switch off and then reduce the amount of glucagon that they were producing. The last thing you have to know about is how insulin secretion is controlled. So the beta cell on the left here, this cell is at rest. It is not currently secreting insulin. We have its potassium ion channels are open, and so potassium ions are diffusing out of the cell, which means that we have a potential difference across the membrane of -70 millivolts. The cell on the right-hand side is now in a stimulated state. We have high glucose concentrations around the outside of the cell, and they are passing in through glucose transporters in the cell membrane. And you can see that in the bottom of the picture, those glucose molecules are passing into the cell through those glucose transporters in the membrane. They are being metabolized by the mitochondria to release ATP. That ATP binds to those potassium ion channels and causes them to close. Those potassium channels are referred to as ATP sensitive potassium channels. So a high concentration of ATP within the cell causes them to shut. As those potassium ions can no longer pass out of that cell, we get a change in the potential difference across the membrane, and it changes from minus 70 millivolts to minus 30 millivolts, and we have depolarization. That depolarization causes those voltage-gated calcium ion channels to open, so of course calcium ions enter the cell and they cause the secretory vesicles to release insulin outside of the cell by exocytosis. Have a look at these statements, which is to do with the control of insulin secretion, and see if you can put them in the correct order. Okay, hopefully you've had a go at that, so let's look at the answers. So, number 1, at normal blood glucose concentration levels, those potassium channels in the plasma membrane of the beta cells are open, and those potassium ions are diffusing out of the cell, and we have that potential difference of minus 70 millivolts between the inside and the outside. When the glucose concentration increases, that glucose starts to enter the cell by glucose transporters. The glucose is then metabolized by mitochondria, releasing ATP. That ATP binds to those potassium channels and causes them to close. Remember, they are referred to as ATP-sensitive potassium channels. As the potassium ions can no longer pass out of the cell, the potential difference changes to around 30 millivolts and depolarization occurs. That depolarization causes voltage-gated calcium channels to open, and therefore calcium ions enter the cell and cause those secretory vesicles to release the insulin by exocytosis.